This study we're presenting today is titled Muscle Fiber Switching via TRPM2 Mediated Calcium Signaling. This study modulates the existence of TRPM2 receptors to try to affect muscle type in mice. The researchers got their hands on TRPM knockout mice where the receptor was removed and control mice. The premise behind the study is that muscle fiber types can switch based on what type of exercise is being undergone. Slow twitch muscle generally changes to fast twitch muscle during strength training, and endurance training generally changes fast twitch muscle into slow twitch. Skeletal muscles have a remarkable capacity to undergo adaptive changes in response to use and disuse, including changes in fiber size, fiber type, and muscle force. The main fiber types being studied here are type 1, which is oxidative slow twitch muscle, and type 2B, which is similar to type 2X in humans. Both are glycolytic fast twitch muscles. You have evidence of glycolytic fast twitch muscles if there is lactate present. The proposed factor that will change the fiber type in muscles is NFAT-C1, that is nuclear factor of activated T cells, cytoplasmic. It inhibits MyoD-dependent fast twitch muscle fiber gene promoters and is active when it is translocated into the nucleus by calcineurin, a calcium-dependent phosphatase. The reason why we are modulating TRPM2 in this study is that it is a calcium-permeable cation channel. These are present in a lot of parts of the body, the brain, the pancreas, the heart, and immune cells, and is generally attached to cell proliferation, insulin release, cell motility, and cell death. The role in skeletal muscles isn't very well understood, but because it is a calcium-permeable cation channel, the researchers speculate that the exercise abilities of TRPM2 knockout mice would be impaired, and their two goals of this study is one, to find out whether skeletal muscle fiber type transition and mitochondrial function were different following exercise training between the two types of mice, and if there is a difference present, what the mechanism is by which TRPM2 mediates calcium signals in skeletal muscle during exercise.
So the experiment was designed by having two different groups of mice, some that were untrained and then some that were trained. I will go over how the method in which they were trained, but there was a wild type and a TRPM2 KO group for both the untrained and trained group of mice. The trained group, the training they went through was on a treadmill, and they would run the mice for 60 minutes for 5 consecutive days in a week, and they continued this process for 4 weeks. And from there, they would also do an exhaustion test where they would run the mice. For the training, they would run the mice at a pace of 12 meters per minute. And in the exhaustion test, they'd run them at 6 meters per minute, and they would measure how often the mice would fall off the treadmill in a span of a minute up until it got to more than 10 falls off the treadmill per minute. But then the mice, after the 4 weeks, were dissected, and muscle analysis was taken of the muscle growth and the change in muscle fibers. And these tests they did were on the calcium levels, the ATP levels, lactate levels, and also the mitochondrial potential that the muscles had.
So TRPM2 knockout mice have fewer exercise capabilities than wild type mice in exhaustion tests. First, looking at the key for these graphs, blue will indicate the non-trained wild type mice, red indicates the non-trained TRPM2 knockout mice, green is the trained wild type, and purple is the trained TRPM2 knockout mice. Looking at graph A, we're looking at the running time for the 4 different groups of mice. Looking at the non-trained wild type compared to the trained wild type, we can see a massive increase from 60 minutes to almost 100 minutes of running time. Whereas between the non-trained knockout, the trained knockout, there's almost no difference whatsoever. Moving to graph B, we're looking at the running distance of the 4 groups of mice. First, again, we can see that the knockout mice for the trained and untrained is almost exactly the same. Whereas again, with the non-trained wild type and the trained wild type, we can see a huge increase in running distance that they're able to achieve. And then looking at graph C, we see the percentage of running mice over time. And again, we can see the green and the blue, which is the wild type, far outdid the knockout mice in the fact that more mice were able to run for a longer period of time than the other mice. In the research, they measured the weights of the skeletal muscles as well as body and heart weight. They did this because they weren't sure if the muscle fiber types were switching or if we were just adding muscle mass and that was causing the increase in running time and running distance. So they took the mice and they measured the muscles and the heart weight, and they found no difference in those between the untrained and the trained. And so they could conclude that it didn't come down to adding muscle, but instead fiber type switching. This next graph, again, it's the same color as blue, is non-trained wild type, red is non-trained knockout, green is trained wild type, and purple is trained knockout. In this case, we're looking at fast MHC, which is fast twitch muscles, and slow MHC, which is slow twitch muscles. We can see that in the first graph on the bottom, in the fast MHC, that the non-trained wild type and the trained wild type, we can see a decrease from non-trained to trained in the fast MHC, whereas we really don't see a decrease. If anything, there's a slight increase between the non-trained knockout compared to the trained knockout. Looking at the slow MHC, we see a great difference here. So the non-trained wild type, the blue, were able to increase their slow twitch when we look at the green trained wild type by almost double. And this is not seen in the knockout. The knockout had almost no increase whatsoever and stayed basically the same, meaning that we weren't seeing transitioning from the fast twitch fibers to the slow twitch fibers. And we can see the same thing here just in picture form when we're looking at the fast twitch muscles in the non-trained wild type and the trained wild type. We see a slight degradation in those. And in the knockout between the non-trained and trained with the fast MHC on that first row, there's almost no difference whatsoever. Whereas in the bottom row, we're looking at slow twitch fibers, and the non-trained wild type to the trained wild type, we can see a huge increase in development in those slow twitch fibers. Whereas looking at the knockout, we almost see nothing happen. It almost looks the same in both images. Next, we're looking at the NFAT-C1 controlling muscle fiber type and switching in response to exercise. The NFAT-C1 translocation, we can see that in the control wild type and the electrically stimulated wild type, which is the blue and green, we have a huge increase in the percentage of NFAT-C1 that's translocating, whereas in our knockout mice, the red and the purple, again, there's almost no difference. And just like what was said earlier, NFAT-C1 plays a massive role in the muscle fiber switching. And so if that's not working, then we can assume that TRPM2 is playing a role in that inhibition. Whereas the NFAT-C1 translocating, we can see from this, the tests that they ran in the wild type and in the control group for the wild type and the knockout, almost all the NFAT-C1 is found in the cytosol, and almost none in the nucleus. Whereas looking at the electrically stimulated mice, the knockout had almost all theirs still in the cytosol, whereas in the wild type, there had a massive amount that moved and translocated into the nucleus, meaning that those fiber types are still able to switch, and NFAT-C1 is inhibited because TRPM2 is not messed with in that control group. Looking at the skeletal muscle calcium levels, blue is our control wild type and red is our control TRPM2 knockout mice. If we look at the levels in the blue graph, we can see that calcium concentrations over time are much higher than if we looked at it compared to the red, meaning that the calcium levels aren't gaining to the level that they need to in order to help that muscle fiber type switch and work correctly. The next calcium levels we looked at is in the mitochondria. Again, blue is for wild type and red is for knockout. You can see during the electrical stimulation time marked by the ES on the graph, the concentration of calcium over time increased significantly in the wild type, whereas in the knockout, it didn't increase as much. And the researchers assumed this was due to the fact that the wild type's muscles had transitioned to slow twitch muscles, which use lots of oxidative ATP synthesis, which would be the mitochondria playing a role in that. Whereas in the knockout, they mostly had fast twitch fibers, which meant that they were using mainly glycolysis and not the mitochondria.
We can conclude from the study that there is a correlation between the TRPM and the switching from fast twitch to slow twitch, as we concluded that the knockout mice primarily had the fast twitch muscles, the same amount of fast twitch muscles and less slow twitch muscles than when the experiment started. One of the things they used to prove this was the lactic acid, or the lactate, the levels of lactate in the skeletal muscles. As we know, for fast twitch muscles, you mostly use glycolysis as a burst of energy, a quick source of ATP. The knockout mice had higher levels of lactate in their skeletal muscles. They also correlated the levels of mitochondrial calcium, mitochondrial membrane potential, and the ATP levels of the knockout mice. As calcium leaves the mitochondria, the membrane potential changes. The membrane potential has to be a certain voltage in order for ATP synthase to produce ATP. They showed that the knockout mice had less ATP than the control type. Something that was also interesting was the correlation between the TRPM and muscle atrophy. So fast twitch muscles seem to atrophy quicker than slow twitch muscles. So a study that they suggested would be interesting was how to follow that throughout one's life, but also that TRPM maintains fast twitch muscle or slow twitch muscle quicker. And they suggested that in a therapeutic setting, whereas if you're not using a muscle or in the elderly folk that don't use muscles as much, that they could target these receptors and maintain their muscular toneness. And this is the study that we studied for this presentation.